Hello everyone and welcome to today's webinar titled HMGB1 to help in the understanding of COVID-19 disease. This webinar is part of the 2021 Microbiology Virtual Week. And I'm Susie Valdez of LabRoots and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by TCAN. For more information about TCAN, go to www.tcan.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive and we encourage you to participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of our presentation. Also, if you have any trouble seeing or hearing this presentation, just click on the box located on the far left of your screen. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located on the top right-hand corner of your presentation window and follow the process to obtain those credits. Without further ado, I now present today's speaker, Dr. Ulf Andersson, a professor of pediatric rheumatology at Karolinska Institute. For a complete biography of our speaker, please visit the biography tab at the top of your screen. Dr. Andersson, welcome. You may now begin.
My name is Ulf Andersson. I have worked as a professor in pediatric rheumatology for many years at the Karolinska Hospital in Stockholm, and at the same time I have performed basic research in the context of cytokines and the last 20 years in the context of HMGB1 and the cholinergic anti-inflammatory pathway at the Karolinska Institute in Stockholm. HMGB1 is probably the most studied molecule in the alarm system. When we talk about alarmins and DAM signals, HMGB1 is in focus. It is a nuclear protein present in all cells. When a cell gets in trouble or threatened or even dying, then HMGB1 will be released as an inflammatory signal. Any lytic cell death will cause HMGB1 extracellular release. The second pathway for HMGB1 to get extracellular is via active secretion, and that happens in many different cells which are activated or stressed. Once HMGB1 is extracellular as an alarm signal, it uses two different receptor systems to induce inflammation. HMGB1 is generally complex bound extracellularly with other molecules like DNA, RNA, histones, nucleosomes, endotoxin, IL-1 alpha, IL-1 beta, and other danger molecules. And these complexes will bind to RAGE. The consequence of this is endocytosis, and the complexes will first end up in endosomes and later in the lysosome system. Normally, lysosomes will get degraded, but the unique thing with HMGB1 is that it works like a detergent in high intralysosomal concentrations. And that will mean that the lysosomes will be damaged. And that will enable HMGB1 partner molecules access to the cytosol and access to sensors for these molecules. And that's the background why HMGB1 may be so dangerous in COVID. talking about respiratory viral infections, I think influenza and RSV virus bronchiolitis are the best studied viral infections. And what has been found is that systemic levels of HMGB1 are detected and correlate to the severity of the pneumonia or bronchiolitis. We know that this is a lytic viral infection in epithelial cells in the respiratory tract. That means excessive cell death, and that by definition means that H1 will be extracellularly released. And then you get secondary inflammation and active secretion by inflammatory cells, adding H1 additionally to the extracellular pool. So H1 must be around in huge amounts in the lungs in severe disease. Another important fact is that it has been shown that the arterial concentration of HMGB1 is only 60% of that found in venous blood in blood samples taken simultaneously. That means that a substantial pool of H1 is retained in the pulmonary circulation. RAGE can be induced in inflammation in many other cell systems, but if we look at it at the physiological situation, then the lungs are the only organ that has RAGE expressed on cells on the surface. So I think that the purpose of this is for RAGE in the lungs to eliminate not HMGB1 itself, but complexes of HMGB1, that is the danger molecules that HMGB1 are complex with extracellularly, like DNA, RNA, LPS, and other unwanted extracellular molecules. They, under physiological circumstances, they will get to the lungs and be then eliminated in the lysosomes. But if you have huge amounts of these complexes and huge amounts of intralysosomal HMGB1, then you get damage to the lysosomes, which may then enable these potentially dangerous molecules like DNA and RNA and histones and nucleosomes and LPS to get into the cytosol. I think this goes both for the cytokine storm and for the clotting catastrophe, which are both so harmful events in severe COVID. I want to add one recently identified factor to this pathogenetic scenario, and that is that patients who have severe COVID, they have detectable SARS-CoV-2 RNA, fragments of RNA in the circulation that can be detected for 3 weeks extracellularly. At the same time, this Oxford group who published the paper could never find infectious intact virions in circulation, only in the respiratory tract. As long as the RNA is extracellularly, it is probably rather unharmful. But the moment that HMGB1 enables this RNA to get intracellularly to the cytosol where there are sensors for RNA like RIG-I and other molecules, then they may very well take part of the pathogenesis. We know that inflammasome activation ends with cleavage of a molecule called gasdermin D, which is a pore-forming molecule. But it has another also important mission, and that is to translocate the phosphatidylserine that is inside the cell membrane to the outer side on the cell surface in an inverted shape. And that phosphatidylserine is a perfect ligand to tissue factor, the molecule that starts the extrinsic coagulation system. The combination that HMGB1 transported molecules will get phosphatidylserine expressed on cell surfaces, and HMGB1 via Toll4 and an NF-kappa B activation will upregulate tissue factor on endothelial cells and on macrophages and myeloid cells. Is then a very dangerous situation that will cause coagulation. I want to point to one fact that has intrigued me a lot, and that is that there are a lot of unusual neurological manifestations in COVID. And one fact is that smokers are partially protected from the severe form of COVID. And it's very likely that it is the nicotine that is the active molecule here. And nicotine acts via nicotinic acetylcholine receptors. And that points then to the autonomic nerve system as being a disturbed system in severe COVID. We know that there is something called the cholinergic anti-inflammatory pathway that is mediated via acetylcholine. And we know that the vagus system is central for this neuro control of inflammation. And the fact that nicotine helps to prevent too much cytokine formation indicates that there is an autonomic dysfunction in severe COVID. The cholinergic anti-inflammatory system does not work properly. Here is really a paradox. I mean, if you check PubMed, you will find around 50,000 hits on if you print COVID-19. But if you try to find studies where they have measured HMGB1 in COVID-19, you will find one such study so far, and that study found increased level of HMGB1. But this is really a paradox. We have tried to use the thickened Schiller test ELISA that is so successful in all other diseases we have tested to measure HMGB1 in severe COVID patients. And the remarkable finding that we have come to is that we don't find increased level of HMGB1 even in patients who have CRP of several hundred units. We find this unlikely to reflect the true biology. I mean, you have massive cell death, you have massive inflammation. We know that these are conditions which always generate increased systemic HMG1 levels. So we suspected that the ELISA antibodies could not detect HMG1 because there were molecules attached to HMGB1. I mean, the way that I have discussed before, that in vivo HMG1 binds so many extracellular danger molecules like nucleic acids, LPS, and other molecules. What we then did is that we used perchloric acid as a very harsh treatment in order to pre-treat the plasma samples to try to dissociate possible molecules that were attached to HMGB1. And that turned out to be the case. When we did this pre-treatment, then we found high levels of HMG1 systemically in patients with severe COVID, despite the fact that this tough pre-treatment also must have damaged some of the HMG1 molecules. So we anyhow found strongly increased HMG1 levels. We have confirmed this by using Western blot analysis too. But there is obviously a major problem here in COVID, that molecules attached to HMG1 and these are so hardly bound that the conventional buffer step that is always used in the Cytokine ELISA procedure does not work to dissociate the molecules. Thank you.